Hello guys, a very good morning to all of you. In my last lecture, I told us about B-cell maturation. So continue this topic. Let's start. I told you B-cell originate in bone marrow. This is bone marrow. For the B cells mature in bone marrow or differentiate in bone marrow. So, in bone marrow, lymphoid lymphoid stem cells differentiate first of all in presenter B cells, which you can say pro B cells. I tell us all of you. I told us pro B cell contains some receptors. One is Ig alpha, beta, and CD13, CD24, CD43, CD45R, c-KIT. All these receptors are present on B cells. And one is stroma cell of bone marrow. Stroma cell of bone marrow. So guys, the stroma cell bind to, this is stroma cell receptor. Stroma cell receptor. At this, the stroma cell of bone marrow binds to this pro-B receptor. When they bind, means after binding, it secretes IL-7, interleukin 7, which further binds to interleukin 7 receptor. Interleukin 7 receptors which are present on pro-B cells. इंटरल्यूकिन 7 रिसेप्टर से जाके बाइंड होगा जो कि pro-B cells पे प्रेजेंट होता है. Further, due to binding of interleukin 7 on pro-B cell, heavy chain gene rearrangement occur and forms pre-B cells, precursors B cell. Fine. After binding, after binding of interleukin 7 on pro-B cell पे बाइंडिंग होने के बाद इंटरल्यूकिन 7 की हैवी चेन जीन रीअरेंजमेंट occur. So it converts into pro-B cell convert into pre-B cell. Pro-B cell converts into pre-B cell, means precursor B cell. Precursor B cell. So similarly, pre-B cell also contains some receptors like pro-B cell. First is Ig alpha beta. Some receptors are CD19, CD45R, CD220, or CD25. These receptors are present on pre-B cell. Pre-B cell किस पे present है ये receptors? Pre-B cells, and there is heavy chain rearrangement हो गया था यहाँ पे. There is heavy chain rearrangement and a V pre-B chain also. Lambda V pre-B chain also, which is known as surrogate light chain. Mu chain means IgM. Okay, and a surrogate chain. Which you know as lambda V pre-B. Okay, which is also known as surrogate chain. These surrogate chain check that whether a light chain is able to attach this point or not. Okay, so there is a light chain rearrangement occur. Similarly, light chain rearrangement occur where pro-B cell, after interleukin attached to the pro-B cell, and it shows heavy chain gene rearrangement, or it converts into pre-B cell. Here, after light chain gene rearrangement occur, it converts into immature B cell. Okay, so due to heavy chain gene rearrangement, mu chain occur on the surface of pre-B cell. Pre-B cell पे heavy chain rearrangement occur होगा, and another surrogate light chain also occur. It also it consists two component. जो लाइट चेन सरोगेट लाइट चेन अरेंज हुआ है, ये कंसिस्ट करता है टू कंपोनेंट्स, लैम्डा 5 चेन एंड वी प्री-बी चेन. ओके, तो सरोगेट लाइट चेन अकर टू चेक ओनली वेदर ए लाइट चेन बाइंड और नॉट एट दिस पोजिशन. Whether a light chain bind or not at this position. So after light chain gene rearrangement, it converts into immature B cell. It converts into immature B cell. Okay. So immature B cell consists Ig alpha beta and IgM. It is a monomeric in nature. IgM are in monomeric in nature. IgM is a membrane bounded antibody. Membrane bounded. Okay, so Ig alpha beta and IgM together called as B cell receptors. अब हम BCR की बात करते हैं guys. So Ig alpha beta and IgM together called as B cell receptors. B cell receptor together called B cell receptors, BCR. Okay, further immature B cell converts into mature B cell. Mature B cells or navy B cells. Okay, a mature B cell consists Ig membrane bounded IgM, Ig alpha beta and another antibody is Ig membrane bounded IgD. These are present on navy B cell surface. These receptors present on navy B cell surface. So there is two antibodies which are membrane bounded are IgM and IgD. Mature or navy B cells leave the bone marrow and goes into circulation and reach to secondary lymphoid organ. Here mature B cells are leave the bone marrow. They leave the bone marrow after maturation and goes into circulation and reach to secondary lymphoid organ. These secondary lymphoid organ are lymph. These secondary organs are lymph nodes, spinels. Further, they reaches to secondary lymphoid organ, lymph node. After reaching secondary lymphoid organ, they interact with antigen and it became activated. After interacting antigen, the B cell lymph node, sorry. After reaching secondary lymphoid or node, it B cells became interact with antigen then activated. After encounter with antigen, it became activated B cells. So B cells activate after entering to lymphoid node where it encounters with antigen. For B cell activation, it requires antigens. Okay. Further, antigens are depending on nature of antigen. It is divided into T helper cell dependent B cell activation or T helper cell independent B cell activation. So guys, T cell maturation is complete. This topic is completed. Overview is that B cell mature into origin into bone marrow and then differentiate or mature also in bone marrow. B cell lymphoid stem cells differentiate into progenitor B cell which consists some receptors and also a stroma cell receptor where stroma cells of bone marrow attach and release interleukin 7. These interleukin 7 attach to the interleukin 7 receptors of pro B cells. After binding interleukin 7, on pro B cells, heavy chain gene arrangement occur on pre B cell. It consists mu chain, which is heavy chain IgM, and also consists a surrogate light chain pre B cell. These surrogate light chain consist two components Ig alpha 5 and V pre B. These light chain gene arrangement occur and convert the pre B cell into immature B cell. These surrogate light chain check whether a light chain bind or not at this point. So, the pre B cells after light chain rearrangement convert into immature B cell. Immature B cell consists Ig alpha and beta and also IgM, which is monomeric in nature. So, immature B cell convert into mature B cell. These mature B cell एक पॉइंट रह गया. IgA alpha and beta and membrane bounded IgM together known as B cell receptor. In competitive exams or any entrance exam, there is examiner asks that B cell receptors. These B cell receptors are IgA alpha beta and membrane bounded IgM. After maturation of B cell, the mature B cell consists membrane bounded IgM and membrane bounded IgD with Ig alpha beta. There is two membrane bounded antibodies, IgM and IgD. These receptors present on navy B cell, or we can say mature B cells. After maturation is complete at this point. After maturation, they leave the bone marrow and goes into circulation and reaches to secondary lymphoid organ. Here secondary lymphoid organ is lymph nodes, spinals. After reaching secondary lymphoid organs, they encounters with antigen. After encounter with antigen, it activated B cell activated. Further, antigen depending on nature of antigen, it divided into activation of B cell divided into two types: T helper cell dependent activation and T helper cell independent B cell activation. So that's all for today. Thanks all of you for supporting me. Guys, if any query, please message me, then I can try to solve all of your queries. Thank you.